And we need to consider the principle known as selective toxicity. You might see this in context of cancer chemotherapy, antivirals, and antibacterials. So what does that mean? Well, obviously it means that you're targeting something in a selective way. So we're trying to exploit, in case of antibiotics, we're trying to exploit the differences, the biochemical and structural differences between the bacteria and the host, which is the human body. And we're selectively targeting features that are unique to those bacteria. And the whole purpose of doing that is we want to minimize the adverse effects. So what are really the key differences, and what are the features that are so different from us, and yet if we inhibit those things or target those things, that will inhibit bacterial growth or kill them. So that's the reason why we need to select antibiotics that don't have any endogenous targets within the body. Now that said, so for example, penicillin will have effects on many bacterial species, but it's not supposed to have any specific target within the human cells. Now that said, with all antibiotics, there's always a potential for adverse effects. So at least one adverse effect is present in pretty much every antibiotic, and that is the adverse effect associated with normal flora. So all antibiotics have the potential, almost all antibiotics at least, have the potential to disrupt the balance in terms of our gut flora. And if we disrupt that, then that could change the situation for the host. We've talked about the supra infection. I talked about how the normal flora is important for normal health. So all antibiotics will have some degree of impact on the normal flora. Let me just change the color. Okay. So before we go on, let's talk about specifically the features that are unique to bacteria. Let's actually compare bacteria and animal cells. And this is probably a review. Prokaryotes are bacteria, so they contain the cell wall. They don't have any nucleus, so their genomic material and other features are not within the defined nucleus. They don't have cytoplasmic organelles such as mitochondria or Golgi, and they don't have endoplasmic reticulum. So their ribosomes are actually floating around in the cytoplasm. And even the ribosomes are different. The animal cells have larger, more complex ribosomal complexes. The two major subunits are 60S and 40S, which make up an entire complex of 80S ribosomal complex. In prokaryotes, that complex is relatively simpler with two smaller subunits that add up to a less sophisticated and smaller ribosomal complex. Reproduction in bacteria occurs simply by binary fission, and in contrast in animal cells we have mitosis. So we have mitotic structures that are associated with different stages of mitosis. Bacteria, very simple, divide and divide again. Now, some targets are enzymes. So we'll talk about targets for sulfonamides. We'll talk about tetrahydrofolate reductase as a target. We'll talk about topoisomerase as a target for fluoroquinolone. So these enzymes are present in animal cells, but they're so different, structurally different. They do the same thing in terms of their biochemical pathway, but structurally they're so different from our enzymes that when you target those bacterial enzymes using antibiotics, they have minimal effects on our enzymes. So even when we have a target that is common in animal cells and in bacteria, those targets are sufficiently different from our cells that it usually is okay to use drugs to inhibit those targets. So a simple a bacterial cell will have chromosomal DNA, and we have chromosomal DNA, but our chromosomal DNA has the ability to organize itself into chromosomes depending on what stage of the cell cycle we're looking at. That bacteria, on the other hand, has just this jumbled up chromosomal DNA. It cannot rearrange itself in any specific morphology that we see in chromosomes. What bacteria do have is an additional DNA in form of plasmid DNA. So these are circular, there are smaller circular DNA fragments. So what are the differences? Well, the chromosomal DNA is relatively stable. This is the DNA that defines a species. So if you talk about, say, E. coli, then that E. coli has genes within this chromosomal DNA that are unique to it and are different from other bacterial species. So this gives the bacteria its identity. But plasmid DNA is a mobile DNA, as we'll see later on, and is important for adaptation. Okay, so if you have a bacteria with fairly stable genome, if the environment changes, it can't change its genome quickly enough to survive and propagate. So it has a circular DNA that can easily be exchanged with another bacteria. So it can derive features that allow it to adapt and survive. And a lot of the genes that we're going to talk about, such as resistance genes, or genes that provide the bacteria the ability to resist an antibiotic, are encoded within this plasmid DNA. So we'll talk a lot about how these plasmid DNA fragments are important. So that basically is, in a nutshell, the genomic distribution of bacterial DNA. Just like people, bacteria come in different shapes and sizes. So we have bacilli, or bacillus for singular, is a rod-shaped bacteria. So E. coli, for example, is a bacillus. And we can also have streptobacillus, where you have these rod-shaped bacteria that stay in a chain. And so what happens is the bacteria divides. So one bacteria becomes two bacteria, so it stays attached to itself. And then this bacteria on this side will divide and stay attached to its daughter cell, and this bacteria divides but stays attached to daughter cell. So it causes this chains. An example of that is Bacillus anthracis. So if you've ever looked at pictures on Google Images or something of Bacillus anthracis, they'll form this really long chain. It almost looks like a cable, because there's actually chains that are made up of rod-shaped bacteria. Bacillus anthracis, of course, I talked about this earlier, also produces a potent exotoxin, which gives it the ability to be highly pathogenic. So exotoxin is, in this case, the virulence factor. Bacillus anthracis also has the ability to form spores, so it can basically stay dormant for a long period of time, and that's why it was able. They were back in the days right after 9/11. Somebody was sending out these white powder, and in some instances it turned out to be this Bacillus anthracis, the anthrax-causing bacteria, and that was basically the spores that are hiding, and once they're in the wet environment, moist environment, they will sporulate and they will begin to come alive and cause infection. And until then, they lie dormant. Now you can also have coccus, or spherical shaped bacteria. So we can have streptococcus, so these are spheres in chain. So you'll have a lot of bacterial species that are named Streptococcus pneumonia and so forth. That's because they stay in chain. On the other hand, Staphylococcus means clusters of spheres. Okay, and these are common, such as Staphylococcus aureus being probably one of the most common bacteria that you will encounter. and then we have others that we'll talk about later on. Now, even though cocci and bacilli are the major shapes, you can have bacteria that are unique shapes. So we have Vibrio cholera that actually looks like a bacillus, but it's actually curved bacillus. So we'll just call it a curved bacteria. So not many of those out there, but Vibrio is the major genus with curved species. Cholera, Vibrio cholera, of course, is responsible for causing cholera, mostly in the third world and developing countries. Then we have a couple of other types that are either spirillum or spirochete. And the difference is that spirillum is a more rigid spiral bacteria, and the spirality doesn't really help with the motion in any way. Usually these tend to have flagella that help propagate this bacteria in the medium. So this is more rigid. An example of that is Helicobacter pylori. On the other hand, spirochetes are a thinner and more mobile bacteria. So in here you have more turns within the bacteria, so it can actually form this corkscrew motion within the medium, and that's how it moves around. And a couple of important bacteria that are spirochetes. One is Treponema pallidum. This causes syphilis. And Borrelia burgdorferi causes Lyme disease. So these are common spirochetes. And Spirillum is less common, Helicobacter pylori being one of them. Now, of course, the other way in which bacteria can move around in the body, in fluid at least, is through flagella. So typically bacteria that has flagella will have more than one flagella, but they can have a single flagella. And that flagella will actually make that bacteria move, propel that bacteria in the fluid by turning. And you've seen flagellal motion associated, for example, with sperm cells, but it's also present in bacteria. Example of that is Vibrio cholera, and the flagella can also be a virulence factor, because if a bacteria can swim upstream, say a bacteria that causes diarrhea, but it's able to swim upstream, that that bacteria is difficult to flush out, and so it will survive longer and cause more likely to cause disease. So this is actually a virulence factor. If it can move, then it can be a virulence factor. Now another virulence factor is Fimbriae. So Fimbriae are these like hair-like projections emanating from the bacterial cells all around, and these actually help bacteria attach to mucosal surfaces, whether it's in the lungs or in the throat or in the bladder. These really help the bacteria attach and basically prevent the bacteria from getting washed out. So that's why these are also a virulence factor. Now, everybody knows about cranberry juice and how you should drink cranberry juice if you have a UTI or urinary tract infections. And sometimes you think that's because cranberry juice is acidic, and so if you drink acidic juice, that will help get rid of that bacteria. But it turns out that as far as cranberries goes, they have special chemicals known as polyphenols, which actually compete with this Fimbriae for attachment and actually disrupt the attachment of this Fimbriae on the mucosal surfaces. So that's the reason cranberry juice is unique. Orange juice will not work as well because it doesn't have these polyphenols that will cause detachment of this Fimbriae from the mucosal surface. Now, there are a couple of other, one feature that let's talk about in this slide is sex pilus, or when you have more than one, we call it sex pili. And this is actually a specialized form of Fimbriae, but it allows an attachment between one bacteria to another bacteria, and it's involved in the horizontal transfer of genes. So certain genes can be transmitted from one bacteria to another in form of plasmid. So plasmid can go from this bacteria to this bacteria. So whatever information, genetic information this bacteria had in that plasmid will go to this bacteria. And then once it goes there, it will become an integral part of that bacteria. So here's a schematic showing that. So you have the chromosomal DNA, and this is the plasmid DNA. So what happens is, first the two bacteria come near each other. There's a sex pilus formed between the two. Now what happens is the donor bacteria has the plasmid DNA that's unwound, and one strand goes into the other bacteria, and one strand stays here. Both bacteria will then duplicate that strand and become double-stranded. So now this bacteria has got all that information that was previously in the other bacteria, particularly when it comes to resistance genes. So for example, if this bacteria is resistant to amoxicillin because it contains beta-lactamase, which is encoded in this plasmid, then once it goes here, this bacteria becomes resistant, even though it used to be sensitive to amoxicillin before. Another important bacterial structure is the capsule. Capsule is actually a slimy layer that surrounds the bacteria. This is outside of the outer lipid membrane in the gram-negative bacteria, for example. The capsule is made up of polysaccharides. So there's a lot of sugar, which makes this fairly slippery and slimy. And the capsule is also responsible for giving us biofilms, which are we're going to talk about those later on in the course when we talk about specific strains that cause biofilms on catheters and things like that. The important function of capsule is to protect the bacteria. It keeps the bacteria from drying out. So if somebody is infected with something and they happen to sneeze or cough, and that droplet lands on the table, but if it contains a bacteria that's capsulated, then that bacteria will stay alive for a long period of time without drying out. The capsule also helps the bacteria evade the immune system, because the bacteria have a lot of features within the cell wall as well as in the outer lipid membrane that are recognized by the specific receptors on immune cells, such as these receptors recognize patterns, and these patterns are hidden by this capsule so that immune system fails to recognize the bacteria that's present there. So as a result of these properties, the capsule is an important virulence factor. So a bacteria with the capsule is more likely to survive and cause a more significant infection than a counterpart that does not have the capsule. Now, capsule is present in many bacteria such as E. coli, Salmonella. So that doesn't mean that all E. coli have capsule. Only the strains that are capsulated are the ones that are more virulent. Same with Salmonella. Now, an important one that I wanted to discuss is Haemophilus influenza. So that's Haemophilus influenza was named because they thought it causes influenza, but of course influenza is caused by the flu virus, so it's not the one. And the reason why they thought was the bacteria, because it also causes a lot of upper respiratory tract infections such as sinusitis, otitis media, and so forth. Now, Haemophilus influenza can be divided into two types. So there's type A and then type B. Type A actually causes milder infections such as sinusitis, which I've already talked about. Type B is more virulent, causes meningitis, which is of course serious. So to prevent this, back in the 80s and early 90s, they developed a vaccine because this was a serious concern in those days. And so we have a vaccine against the type B, which is Hib. And it's actually type B, which is capsulated. And it's the more serious type. So if you're vaccinated with Hib, then you are safe from the more virulent strain, which is the capsulated strain. But type A is still around, and it causes milder infections, but those are manageable. It's the meningitis that's more serious. So here's an example of how a capsule differentiates a virulent strain from a non-virulent strain.